Okay, this morning, Gillian and I are going to be talking about cancer for the first hour. I'm going to take about, I hope, only 15 minutes to introduce it, and then Gillian is going to talk about reproductive cancers. So I'm going to have basically 5 things to say about cancer, and I'll summarize them on the last slide. But I'd like to just start by reviewing the medical implications of cancer. The lifetime risk in the U.S. that somebody will get cancer is about 1 in 3, and as we live longer, that will go up. So if you're thinking about things to extend your lifespan, just realize what will greet you at the end. A lot of women get breast cancer. If we do autopsies on people that are dying of other causes like war or traffic accidents, we discover in them that they have many tumors. And that indicates, in fact, that we are all initiating tumors and that our immune system is killing them. It's actually probably the rare cancer that makes it through to diagnosis. It takes about 1 million cells in a mass before it can be picked up in diagnosis. And 1 million cells is already a long individual clonal evolutionary history. If we all lived long enough, probably all men would get prostate and all women would get breast cancer. And the only saving grace, the silver lining in the cloud, is that the older you are when the cancer starts, the more senescent the cancer is and the slower it grows. Now, the first take-home message is this. Stem cells are a double-edged sword. Cancer only happens in multicellular organisms. And multicellular organisms invented stem cells for repair and differentiation. At the outset of multicellularity, the distinction between soma and germline was established. And the germ cells, which you can see here in Volvox is the bright green, are going to make the next generation, and all these other guys are feeding them. They do that because they're 100% related to them. But if there is a mutation in one of the somatic cells, it's no longer 100% related to the germ cells, and there is an evolutionary pressure for it to go off on its own and do its own thing. So how is the covenant enforced? Well, one is the physical isolation of the germline, which in many organisms happens early in development, but interestingly in some invertebrates does not. In fact, it's possible for, quote, somatic cells to get into the germline fairly late in development in some invertebrates. That's the kind of thing you could study here at this lab. But there are tumor suppressor genes, and they are actually patrolling the body and suppressing tumors. They are checking on cells remaining stably in their differentiated state, and if a cell starts to proliferate when it shouldn't, the immune system tells it to kill itself. And I really like this quote, every cell wakes up in the morning thinking about committing suicide, and it has to be talked down by its friends. Okay? It's kind of a, oh my goodness, that's all of me. Most cancers originate in stem cells, and they're a great innovation. They divide slowly, and they retain the potential to differentiate. Their slow division reduces cancer frequency, but their potential to differentiate pre-adapts them to a cancerous lifestyle. That is an epigenetic state. It's not a mutation, okay? So the population of cells in our body that is forming the origin of most cancers is epigenetically different, but not genetically different from all the other cells in our body. The stem cells that are positioned to replace cells that wear out are in bone marrow, lungs, intestine, and skin, and my goodness, aren't cancers of bone marrow, lung, intestine, and skin common? And aren't cancers of heart and things like that and muscle less common? Okay, second message. Cancers result from the accumulation of mutations, and there are a lot of mutations. So this is actually Steve Frank's analysis. During development, we expand to about 10 to 100 trillion cells. That's how many cells we have in our body. That requires one less than 10 to 100 trillion cell divisions. And at each division, the mutation rate per gene is about 10 to the minus 7th to 10 to the minus 6th. That means that the number of somatic mutation events per gene per individual is 1 million to 100 million. Each of us has had between 1 million and 100 million mutations in every gene in our body as we grew up. Now, those mutations are coming from bad luck. Those are mistakes during mitosis. They're coming from mutagenic exposures, UV. They're coming from chronic wounding and inflammation, which increase the concentrations of protons in our tissues. And the kind of exposure that results in mutations is, the worst kind is chronic. An acute exposure is not nearly as bad as steady daily exposure at a lower level. One bad sunburn isn't as bad as being out in the sun every day for 20 years. Now, this is what a molecular biologist sees when she looks into a tumor. This is a sample from lung cancer. The blue is a cell nucleus. Chromosome 17 is marked in pink. Chromosome 11 is marked in green. You ought to have 2 of them. This cell has 1 copy of chromosome 17, and it's got about 10 or 11 copies. This nucleus has 10 or 11 copies. And you can see that, in fact, one of the things that goes on in cancer is that they become a genetic wilderness. They become genetically extremely diverse. And the reason for that is that some of the key mutations are mutations that increase the mutation rate. Now, why does cancer get nasty? Why does it get virulent? Well, it doesn't need an external signal to stimulate its proliferation because it's ignoring signals to shut down proliferation. So some of the key mutations are the ones that say, buzz off, I'm not going to listen to the immune system tell me to kill myself. And other key mutations are the ones that increase the mutation rate because it's going to have to get 8 or 9 in sequence. Cancer cells can divide indefinitely, and they do that by stabilizing their telomeres. So mutations in telomerase are another important thing in cancers. They do have these increased mutation rates. They can evade the immune system. They can induce angiogenesis. And probably any proliferating mass in the body has that capacity. That's probably not a mutation. That's probably something that all cells have in them, that when they are differentiating and proliferating in the body, they are going to be sending out, normal healthy cells will also be sending out signals to attract vascular growth. And then they are able to actually move and disperse and colonize new parts of the body. So that's metastasis. They are evolving through natural selection. So I'm giving you messages out of evolutionary biology that come both from deep time and macroevolution. Stem cells predispose us to cancer. And I'm also giving you a message from microevolution, which is that every cancer undergoes a microevolutionary process in our body. We can go back to our initial conditions. Do they have genetic variation? Yes, they are a set of genetically different clonal lineages. They are competing with each other for host resources, and the competition for host resources results in the ones most capable of extracting resource from the host and most capable of colonizing new areas in the host. So their variation, their genetic variation, is actually affecting the reproduction and survival of the cell lineages, and they are doing this because they can ignore the signals to shut down proliferation. So to think about that for a minute, this picture, which I hope becomes informative to you, is an important one because it shows the difference between asexual and sexual evolution. These clones that are evolving in our bodies are reproducing asexually. The top picture is from Crow and Kimura, 1966. It's a classic picture in evolutionary biology. And what it shows you is you could imagine, say, that A, this light purple up here, is normal tissue. We'll go through that with Barrett's esophagus below. In order to get full malignancy, you might need the combination of A, B, and C. So 3 mutations in 3 different genes. Okay, so one clone gets B, but it doesn't have A. A is an advantage and will allow it to spread. One mutation gets B, it dies off. One clonal lineage gets C, it dies off. That's what those little blobs on the left mean. Then along comes a C mutation in a lineage that already has A, and it starts to spread a little bit, but another member of that clone, another cell lineage, gets the combination A and B in sequence. It's better than AC, it outcompetes it. This one goes extinct, this one spreads. And then finally, after this is established, millions of cells, the C mutation occurs in a clone that already has A and B. That was something that Crow and Kimura did just for asexual evolution in general, and it's making the point that asexual lineages have to acquire mutations in sequence. They can't trade information because they don't have sex. There's no way to move with horizontal gene transfer among those lineages and get the ABC popping up all the way down here. It has to happen one after another. So a cell lineage has to build up to a fairly high number before it's probable that you'll get another mutation in that cell lineage. That's why it takes time. Now this is Carl O'Malley's picture. He just took that and he did it for Barrett's esophagus. So this is the extent of Barrett's esophagus here, and the pink shows just nice, acid reflux irritated tissue. And then you get a neutral mutation popping up and going extinct. Another neutral one pops up and goes extinct. Then in P16, which is one of these key genes that is usually a precursor to cancer, you get one hit, and that one starts to expand. In that clone, there's a neutral mutation. It goes extinct. You get a P53 mutation. It can't outcompete the P16 mutation. It goes extinct. Then you get a double hit on P16. It expands. A neutral mutation pops up. It goes extinct. You get some high-grade dysplasia popping up. It goes extinct. And then in the lineage that had a double hit on P16, you get a P53 hit. And out of that lineage, you get the malignant cancer. Okay? Happening in sequence. And to some degree, we know which genes are doing this. So here is a very nice example that just came out, I think it was earlier this year or late last year. This is for pancreatic cancer, and it has actually many of these processes in it. We start off with a normal duct epithelial cell in the pancreas. The black arrows are mutations in clonal expansion. The red arrows are dissemination to distant organs. There's a whole long sequence, like the one you just saw on the previous slide, that lasts probably about 11 or 12 years, where that kind of thing is going on, okay? And then here, where on the previous slide we finally had the P53 mutation in that other background, you get the parental clone for the metastases, okay? So then you get subclones with metastatic capacity. At this point, those subclones get up to about the million cell level where they can be detected. The patient notices a problem. The doctor diagnoses pancreatic cancer. The diagnosis is too late, and it is coming at about 18 years, 18 and a half years after the process started. The different clones have dispersed through the body, and by looking at their genomes, we can make a pretty good argument that they have adapted to the kind of tissue that they've moved into. So we not only have a splitting of clonal lineages and a dispersal into the body, but we can demonstrate that they're adapting to the local tissue. We've got the whole process of natural selection going on in the cancer, okay? This was where the problem was found. It was found in the liver. This is done with molecular phylogenetics that allows us to reconstruct the individual history of the cancer. Okay? So what you do is you sequence all of these metastases and then apply molecular phylogenetics to it, ask at what point in the past did they have common ancestor, and that's where you get these estimates of times. I think that's very informative. Actually, it's very hopeful. What it does is it tells us that if we can develop very sensitive techniques for picking up perhaps key features of this process, we'll be able to diagnose much earlier. Yeah.
The earlier premise was that these typically originated in stem cells, but the molecule showing us are starting in differentiating cells.
It's in a normal duct epithelial. This one started in a normal duct epithelial cell. Okay? It takes a lot of work. This is sample size of one patient. If you did this on 1,000 patients, my guess is you'd pick up a lot of stem cell. But this one happened to be just a normal duct epithelial cell. Okay, so I want to turn it over to Gillian. My take-home points are, first, stem cells are a double-edged sword. Cancer results from the accumulation of 8 to 9 mutations. Key mutations and inflammation increase the mutation rate, so that once you get a clone that has an increased mutation rate, it's more likely to accumulate the 8 to 9 mutations. Natural selection is what's operating on clones to produce virulence, and the histories of individual cancers can be reconstructed. So, Gillian, I'll take questions while you get set up. And I'm going on again later, so I'll just leave mine up here. Yeah.
That's very interesting, because do each of those mutations then, it's likely that they may have different causes and therefore different implications about prevention? That is, there may be a series of fact features which have their own terms.
Yes. In fact, I think this afternoon you're going to see some presentations on the relationship between things that cause inflammation and their association with cancer that you might not have expected to see. But certainly cancer is certainly not only, but largely a G by E effect. So if you are in an environment like smoking or ultraviolet light or taking particular chemicals in your diet, you're going to have a higher risk of cancer. And that's actually just because of random hits increasing the mutational frequency in a particular tissue.
It seems like there must be a really large genetic component to cancer susceptibility.
There is a genetic component to cancer susceptibility in things like the BRIC1, the breast cancer gene, for example. If you inherit that, then you're already 1 or 2 steps down the pathway. You need the other mutations to get it, but you've already gotten through some of the early ones. And people who have inherited a defect in P53 will have a heightened mutation rate in their somatic lineages and are at great risk. P53 is a repair mechanism.
I heard you say something I've never heard before, so I want to make sure I got it right. That is that some mutations may increase the likelihood of subsequent mutations?
Yes, a P53 mutation is actually a mutation in a DNA repair gene, and if you knock out the ability to repair DNA, then the probability that you'll have a mutation at every mitosis goes way up.
So Steve, you're right in that cardiac and heart cancers are so rare, but the flip side is with myocardial infarction, which is so common, we can't replace our cardiomyocytes. Is that an evolutionary trade-off?
Yeah, yeah. It sure is. We've decided not to have, we don't need stem cells for replacement so much in the heart tissue as we do elsewhere.
Except in the modern day environment.
Except in the modern day environment when we live long enough and we have the G by E interaction in our diet, so we have more animal fat in our diet and stuff like that. You all set, Gillian? Can I give you the mic?
Okay, can you hear me? Yeah? Can you hear me at the back? Yeah, higher. Okay, well, hold it. Okay, I'm going to take a somewhat different tack here on reproductive cancers and look more at the population level, and I'm going to focus primarily on the two most common reproductive cancers, breast cancer and prostate cancer. And I'm going to give you a case study which comes from my own research with Bangladeshis, because there are a lot of interesting things that are going on with this population once they migrate. So just to give you a brief outline of the talk, and I thought it would be quite useful to follow a structure that's analogous to what we've been talking about during the week, which is sort of identifying a problem, framing a question or hypothesis, looking at evolutionary explanations, and in this case also I want to talk about clinical practical implications, which is really important when we're trying to make the leap from evolutionary explanations to some kind of impact for medical doctors. Okay, so in this case, the question that I'm asking about the population that I've been looking at is this one that Randy's raised in general: why are we vulnerable to diseases, and in this case to reproductive cancers? I do want to point out that even though I'm talking about this research, it's involved a lot of collaborations along the way, so I wanted to just acknowledge my collaborators and my students up front, because I certainly haven't been the sole person involved in this. Okay, so let's get some stats out the way first. Of these, I've listed up the main female and male reproductive cancers. Breast cancer, these are the statistics I should add from the UK primarily, but very similar in the USA. So 30% of all cancers for breast. The others are statistically much less significant, as you can see. Cervical cancer, only 2%. Male cancers, prostate accounts for 25% of all cancers in the UK currently, and again, very similar in the USA. So these are, major proportion of all reproductive cancers, as you can see here, and accounting for basically most of the cancers to which UK population is susceptible to. So quite significant. And what are the different causes that can explain why we get these different reproductive cancers? And I just threw up the main ones that we can think about. Physiological inflammatory processes, which certainly go on in breast tissue, because breast tissue is turned over every month as a woman goes through a menstrual cycle. Viral causes for things like cervical, penile cancers. Developmental congenital issues, which can affect things like testicular cancer. You have a 14% increased risk of getting testicular cancer if you're born with a condition like hyperspadias, where the urethral opening isn't in the normal place. Genetic risks, which Steve talked about here. BRCA mutations, which account or give you a 50% higher risk for breast cancer. And then epigenetic environmental issues, which can trigger cancers, and then lifestyle behavior mismatches, and I'll focus on these to a great extent in this talk. Okay, now the problem that we're faced with, as I showed you in the other slide, prostate and breast cancer not only account for the most prevalent cancers in the UK and USA, but rates have been rising, as you can see here. Lung cancer is also very important, so that's up for comparison. But the changes in the incidence of breast cancer and prostate cancer are very worrying. Rates are rising a lot in the Western industrial world. Oops, wrong way. And in particular, if you look at South Asian populations, South Asian referring to Indians, Pakistanis, Bangladeshis, you can see from the top slide that while the incident rate, or the odds ratio of getting cancers, they're below 1 here, is lower in comparison to the white UK, European origin population. The incident rate, the rate at which these cancers are rising in the population in the UK, is much higher for South Asians. So they start off, in other words, from the top slide, at being at reduced rate overall, but once they migrate, they're getting breast cancer, prostate cancer, much a faster rate than the white native population. So that's breast cancer here. The same is true of prostate cancer. Overall, the rates are considered to be low when populations first come into the UK, but the risk is rising. They're getting prostate cancer at higher rates than the white population. So this is a real problem. It's a clinical problem for the National Health Service and for public health in the UK. So let's just first walk a little bit through breast cancer risks and what the main exposure risks are for women, and if we can look at some of these factors and try and figure out if there's anything that we can do if we understand these risks. And the biggest risk for breast cancer, outside of genetic susceptibilities, if you don't have particular genes that make you more susceptible, exposures are really important. And that depends on the exposure to particular reproductive steroid hormones throughout your lifetime. So levels of hormones are important to begin with because we know there's a lot of variation between populations in levels of estrogen and progesterone. The length of the reproductive lifespan, which in women would be defined between age at menarche and age at menopause, is important because if you have a longer reproductive lifespan and you're menstruating for much of it, which is true for contracepting Western women, then you're going to have a longer period of time throughout your life when you're exposed to hormones, and those hormones are turning cells over in your breast, they're turning cells over in your uterus. So it's that process that makes you vulnerable to the chances of getting breast cancer. Hormonal contraceptive use. Although if you use hormonal contraceptives, it's known to reduce your risk of endometrial cancer and ovarian cancer, there are some data that suggest that breast cancer risk may be slightly increased in at least pill users. So that's something else to think about, exogenous hormone use. Fertility, the number of times that you might be pregnant, in which case your reproductive axis is going to be quiescent, so you're not going to be exposed to hormones, levels of hormones. That's very important. Breastfeeding patterns, if you breastfeed your kids for long periods of time, it suppresses the hormonal reproductive axis, so then it reduces your risk of these hormonal exposures. Another thing to think about is postmenopausally, when often women are at higher risk for cancer, partly a function of age, of course, because age, which is at the top, is really important, as Steve pointed out. But if in postmenopause in particular you're overweight, your adipose tissue is producing another variant of estrogen, which can then also increase your risk. And in terms of breast differentiation, we know now that if you are pregnant at a young age, your breast tissue will be differentiated. In other words, it matures enough so that you can produce milk in your breast ducts. If you have an early first pregnancy, that differentiation happens early. It's a significant factor in reducing breast cancer risk. And there was an interesting paper, in fact, in 2005, in which experimentally some clinicians gave young women human chorionic gonadotropin, or HCG, which is the hormone that's produced by a developing embryo. It triggered breast differentiation, and it's thought that this is going to reduce the risk of breast cancer in these women. So there's some interesting stuff out there about potential prophylactic measures that we can use to reduce breast cancer risk. Other lifestyle factors we know are important: diet, partly, or mostly, because probably it reduces your risk for overweight, which is a risk factor. Exercise, we know, suppresses reproductive steroid hormones, so that's really important. Shift work, which affects various hormones. If you do night shifts, you're at increased risk for breast cancer. Environmental exposures, toxins, those may be significant factors too. So I want to throw these general factors out there and then look at them more specifically in relation to immigrant Bangladeshis. Risk for prostate cancer. We know a lot less, actually, about risk for prostate cancer, other than that it's thought that exposure to levels of testosterone are important across the life course. Patterns of sexual activity, men who are in, who are have a lot of sexual activity or who actually masturbate and ejaculate, they're at low risk for prostate cancer. Age at first birth or marriage, I put that up because then obviously one is presumably, not always of course, but exposed to regular sexual intercourse, number of sexual partners, and then things like diet and physical activity are probably important too. So I'm going to talk about my case study then, and migrant Bangladeshis. And the reason why it's become important in the context of these two cancers is because we have a wealth of hormonal data that we've now collected on this population, and we have a wealth of information about their changing lifestyle, changing diets, and so forth when they come to the UK. And we think that what we're uncovering in our research may be very important in explaining these changing risks of contracting breast cancer and prostate cancer, and therefore that has implications for public health. A migrant study, of course, is a really good model to look at some of these issues if we think that behavioral lifestyle factors are important in influencing disease risks like breast cancer. We can rule out genetics if we've got a migrant population, and we're also going back to the population of origin, or at least we can control for genetic factors. And we can also look at short-term effects in these migrant populations. And we started this project primarily just to look at variation in reproductive hormone levels and what happens during early development if migrants come to Britain. But then as a corollary of that, we have been able to extend our work to think about the implications for health in other areas. So why Bangladeshis? Why did we pick on this group? They're actually a fantastic model for being able to control for a host of different environmental and other variables. In Britain, Bangladeshis have been migrating there for well over 100 years in different numbers. Most of the migrants come from one very particular geographical area in Bangladesh in the northeast. And when they come to Britain, at least the London populations that we've been looking at live in quite distinct neighborhoods as well. So you can control for these different areas of geographic origin. I do want to stress as well that we work with middle-class populations. I can't stress that enough because people assume that Bangladeshis are always poor and come from lower socioeconomic background. The guys that we work with, as I've mentioned earlier, they come from land-holding families. In their home country, they can afford servants, including chauffeurs. It's just when they move to Britain, obviously because of exchange rates, they're at the bottom of the socioeconomic scale. In terms of the model that we've used in the different groups, what's important here, there's a lot of information, but basically why are our studies important is because we include first-generation migrants, second-generation migrants that came at different points during their lifetime, including as children, and we have a reference group of white Europeans. We go back to Bangladesh, look at people there, and we've run a number of different projects of women and males of different ages and an ongoing project with kids. We've collected a ton of data which can help us in this reconstruction. We've got biomarkers for hormonal analyses, including blood and saliva, battery of growth measurements, questionnaires which include information about reproductive histories, migration histories, health, nutrition, lifestyle. So we can put together a really nice composite picture of what's going on in these groups. So just to remind you then, after giving you that background to the study, the important exposure factors, including the hormones that we're dealing with, let's go on to actually look at what we found in this Bangladeshi population. So first, one of the things that we found is depending on where you grow up, you will have a major effect on levels of your reproductive steroids. So this graph here is looking at the second half of the menstrual cycle and looking at levels of progesterone. This would be roughly when ovulation takes place. And normally, the pattern for progesterone looks like a normal curve. So that's roughly what you've got here. But the important thing is that black line in the middle, which is the dividing line, if you like, between levels of hormones for those who grew up in Bangladesh, who are the guys, the women below that black dotted line, and above that, we've got child migrants who are the green level, second-generation migrants who are the purple, and then the yellow is the white European women. And you can see that there's a real major difference, it's statistically significant, between where you grew up. So clearly there's some influences of the British UK environment that will make you, when you reach adulthood, have much more robust, higher levels of progesterone, which is a significant risk factor across your life course for getting breast cancer. This second graph down here shows what happens if you migrate as a child at different ages. If you migrate before the age of 8, you are going to have higher levels than if you migrate as a child at older ages. So this migration pattern, and when you do it, is really important for potentially affecting your risk for breast cancer. On the right-hand side, up in the top there, we have some data from one of my students, Keson Majid, who's been working with Bangladeshi males. And again, I put in this black line. The levels, this is showing salivary testosterone, which we've sampled at 3 different times in the day, morning, 30 minutes later, and then at bedtime. And the top 3 lines are high-status white males, second-generation Bangladeshis, child migrants, and then below that, Bangladeshis who still live in Bangladesh, adult migrants, and low-status white males. But again, the point being here that if you migrate early on in life or you're born as a Bangladeshi in Britain, you're going to end up with higher lifetime levels of testosterone, which we assume is going to affect your risk for these highly prevalent reproductive cancers. So that's your general exposure. Okay, this is just a general slide talking about the point that we were very interested in when we started the study, which is how the developmental environment then influences your levels of reproductive hormones. And although this is a case study of Bangladeshis, this may very well apply to other migrant populations too. We did a couple of pilot studies with a colleague, Martin Widgwender, at UCL, because we were very interested in what mechanisms could explain these changes in reproductive hormone levels in these Bangladeshis, given that we know that we're looking at the similar, the same genetic group of origin. So we did a targeted study of estrogen receptor genes and looking at methylation patterns, so an epigenetic pilot study. Using 20 women each in first and second generation Bangladeshis. It's very expensive to do this research. We didn't have a whole lot of money, so that's why the pilot studies are so small. But we did find significant differences between first and second generation women in the progesterone receptor gene and one other estrogen receptor gene on the right-hand side, which is intriguing, but without being able to pursue these studies, it's just intriguing at this point and something worth following up. But I did want to point out, though, that it could be that something is happening epigenetically when these women move to Britain, and it's something that we want to study further. Okay, in terms of early maturation, so thinking about the reproductive lifespan, we know this is important in terms of exposure across the life course to levels of testosterone and the reproductive steroids in females, estrogens and progesterone. So Kesson, my student who's been working with Bangladeshi males, it's very difficult to reconstruct age at pubertal maturation in males. You don't have a clear marker like you do for females with menarche. But he did collect data from questionnaires on 4 different variables which can indicate when puberty happens in males: voice breaking, facial hair shaving, pubic hair development, nocturnal emissions. And he produced a composite. This is, of course, recall data from men. He produced a composite to try and reconstruct age at pubertal development in our different groups of migrants. So here we've got what we call the 7Ts, guys who are still back in Bangladesh, adult migrants, child migrants, second generation males and Europeans on the right-hand side, and it looks like we expected it would, that if you're a child migrant or a second-generation migrant, you mature earlier than if you live back in Bangladesh. So again, this would suggest that you're pushing earlier the age at which you mature, and therefore males would have a longer period of exposure to higher levels of testosterone. For women, we've also got recall data on age at menarche, and age at menopause. And in this top slide, we've got recalled age at menarche from women. If you're a Bangladeshi who still lives in Bangladesh, even if you're middle class, your average age at menarche from these reconstructions is 13 years. If you're a white woman, interestingly enough, we did try and match for socioeconomics. So this is low status white women. Age at menarche was very similar at 13. But look what's happened with the child migrants who came before puberty. The child migrants in the green have an age at menarche at 12. This is statistically significantly different from the other groups. So puberty starts earlier. There's some accelerated growth that's going on here, we know from our anthropometric data. So you've pushed back earlier the age at which you are being exposed to reproductive steroid levels. Age at menopause, we have recalled age at menopause from 2 different studies. And we know that age at menopause in the child migrants is much later than if you are in select, a difference from between 47 years to about 49 years. And again, this is significant in our second study where we've looked at this. And to complement these data of recalled age at menopause, because you can criticize recalled age, women don't remember exactly when it happened, we've done a cross-sectional study where we've taken blood from women and looked at 3 different hormones that tell us about ovarian reserve. And ovarian reserve is the number of eggs that you estimate are left in the ovary, which is going to determine age at menopause. And these 3 hormones tell us pretty much the same thing as we have from the recalled age at menopause. Looking at different age categories here, levels of inhibin, B, and anti-Mullerian hormone, which is that second graph, they decline as women get older. So in these different categories, if you're a Bangladeshi woman who lives in Bangladesh, or you're an adult migrant, your levels are lower. If you're a child migrant or or a European here, then your levels are higher. So, and it's reversed in that last slide because that's follicle stimulating hormone, which is produced from the pituitary, and it gets higher as you approach the menopause. It's one of the diagnostic hormones for menopause. So we have nice confirmation from our hormonal data that menopause is occurring later in these women. So not only do you have an earlier age at menarche, you've got a later age at menopause. We are extending, or these Bangladeshi migrants, their reproductive lifespan is being extended by quite a number of years. We did a multiple regression, which is at the top, and basically looking at all these hormones that we have, the difference between our groups is statistically significant. So this is really important as a changing variable for these women who've migrated. Some of the other behavioral factors, age at first pregnancy, which is in that top slide, there's actually no significant difference between the groups. And then fertility. This is, again, sort of, it's not completed fertility in all our groups, so it's difficult to get much out of it. But fertility is high for Bangladeshi women. Strangely enough, even though the groups of women that we work with are of high socioeconomic status, they're quite conservative religiously. It's a Muslim population, but much more secular than many other Muslim groups. But fertility is quite high, 6.1 in Select. It does tend to lower a little bit in Britain, so they are cutting down on the number of kids, but not by very much right now. The second generation group, who are embedded more into British culture, expenses of schools and so forth, their fertility, we suspect, will get lower. But this is really very striking, our breastfeeding data. Lactational patterns are really important for determining breast cancer risk. In Bangladesh, these women breastfeed for an average of 16 months. Once they get to Britain, it just falls off phenomenally. We're doing more work on this to find out why this happens. It's not as low as well, by the time you reach second generation, it's comparable to the white women, but this is hugely significant. And so it means that one of these lifestyle risk patterns is really changing dramatically. Body mass index, once these migrants get to Britain, you can see they get fatter. This is going to be also an increasing risk factor. And then diet. What's happening to the diet once you move to Britain? These are data from Kesson that we've reconstructed recently. There are changing patterns. Although Bangladeshis tend to like their South Asian diet, they do, things change once they get to Britain. So at the top, for example, rice consumption. On the left are the people still in Bangladesh. Next to that is the adult migrants. Look at how rice consumption falls off as people migrate into Britain. Fruit and vegetable consumption changes. And very importantly, meat, that middle right-hand column. Once people come to Britain, meat, chicken is much cheaper in Britain comparatively than in Bangladesh. So these young migrants and British-born Bangladeshis are consuming a lot more meat than they used to do. So we suspect that this is important. Exercise, interestingly enough, because these Bangladeshis are very wealthy comparatively in Bangladesh, when they get to Britain, they actually exercise more, at least in London too, where many first-generation Bangladeshis can't afford to buy a car. So they're getting more exercise, which is a compensation. So what are the public health messages from this? We can say that rising hormonal levels, as well as these changes in behavior, are going to really have an impact on reproductive cancer risks in these Bangladeshi groups, and we think that public health and policies and health education should try and address this with these populations to point out that their risks are changing and to try and think about some of the behaviors and lifestyles that might mitigate these risks in the future. For men and prostate cancer risk, one of the other things that we did, because we got a little bit of money from a charity called Prostate UK, is my student Kesson administered a questionnaire called the International Prostate Symptom Score to look at lower urinary tract symptoms in men, which is often the indication of benign prostatic hyperplasia, which is thought to perhaps be a risk precursor for prostate cancer. And as you can see here, these symptoms go up again amongst these second generation and young migrants. So we do have some, good questionnaire data from this, an internationally recognized questionnaire that again can sort of help us try and get these health messages out to this population with increasing disease risk vulnerabilities. So those are the data. So I thought I would return here to how we can embed these data into a more evolutionary framework as well. And one of the evolutionary frameworks that's been used in talking about cancer risks is the concept of mismatch that you've been introduced to now a number of times during the course of this week. But one thing that hasn't been mentioned, which I think is quite interesting, because as anthropologists, we talk about this quite a lot, at least in teaching, is not just a mismatch between environments like the Bangladeshis move from in Bangladesh to one in Britain, but there's also this concept called the environment of evolutionary adaptedness, which people argue about a lot because dates are uncertain, the concept is a little bit uncertain, but in general, it's used to indicate the environment in which humans would have evolved early on, the sort of Stone Age diet, if you like. People talk about Paleolithic, Stone Age environment. People talk about Paleolithic diets, Paleolithic environments. But it's thought that there is this disjunction between the environment in which humans spent most of their evolutionary history, this EEA, and the one in which we now live in the West, industrialized and so forth. And one of the explanations for reproductive cancers in general is that we have this lifestyle in terms of diet, reproductive exposures, behaviors that is markedly different from the one in which humans evolved, and which can explain a lot of the risks that we now have to live with. Fertility is part of that EEA environment, higher fertility in women than the 2.1 average in, say, the UK or the USA. But it's also an environment in which your development, as Peter Gluckman pointed out earlier, would have been an anticipatory one, and a correct anticipatory one, of how you would live as an adult. And now with migration, with changing environments that happen so quickly, that there is this disjunction between where you might, your developmental environment in utero or as a child, and the one to which you might be exposed as an adult. So you can look at this mismatch across different levels. I also found this on the web, and I thought it was quite interesting to throw this up here. It didn't copy over very well, but there's some studies out there which claim, or at least this was picked up by the press, cancer is a man-made disease, and I think this ties in with this whole concept of where we are today and where we used to live. Of course, it's nonsense that cancers didn't exist in our earlier environment. Cancers, as Steve pointed out, take a long period of time to incubate. It's very difficult to find evidence for this in past populations, but we do have some evidence. And it's likely that cancers that did exist in the past were just different from the ones that we're vulnerable to today. So let me just go through this. So for example, even though lung cancer we think of as a huge risk factor today because of smoking, it's quite likely that in past times lung cancer was a huge risk factor as well, because people were cooking over open fires. Women in particular may have been vulnerable. It wasn't until chimneys were invented that people had to, could live in houses that were relatively smoke-free. So lung cancer could certainly have been a risk in the past. Stomach cancers, maybe from consuming or chewing on products like cat, betel nut, which are common cultural practices. Liver cancers from things like contracting hepatitis. We don't know what the rates of that were in the past. Oral cancers, again, from chewing things like cat, betel nut, bladder cancer from diseases like schistosomiasis. So of course there were cancers in the past. It's just that the types of cancers that we're exposed to are different. And the risks, environmental risks, are different. But I think we need to point this out in general. And then going back to this concept of mismatch, Randy and George Williams mentioned this very early on in their book, Why We Get Sick, and the British version, I don't know why they changed the title, but they did, Evolution and Healing. And I just put this quote up from the book: "From this mismatch between our design and our environment arises much, perhaps most preventable modern disease." And that concept, as I mentioned earlier, as if you heard from Peter, has been extended to embrace this whole area of research, developmental origins of health and disease, which includes both the developmental environments as well as these other broader concepts. And in this case study that I presented to you, the Bangladeshis are fitting into more this concept of how their developmental environment back in Bangladesh is at mismatch with what they come to once they move to Britain, and that has obviously impacted on their risk for reproductive cancers. So I'm going to finish there with a little bit of time for questions and just throwing up our various funders here. So thank you. Questions? Yeah. Should I give you this, or shall I? I'll just repeat your question just to save time. Yeah, I'll repeat it.
So you got a lot of interesting information. I thought your information on menopause changing in the Bangladeshi who grew up in UK was interesting. So they matched the UK people who live there. So what makes the menopause occur earlier, do you think, in Bangladesh, or is it a different way later?
Right. So the question is, what's going on between the women in Bangladesh, the environment that would cause them to have an earlier menopause? It's interesting. We went round and round this right at the beginning of the study because, as I said, they're well-off people. But what we do think is important, and we're collecting more data on this now, is immune function and disease exposures in Bangladesh. Because even though our populations are middle class, the health system in Bangladesh is really appalling. And the diseases to which you're exposed are many more than you would be exposed to in Britain. Life expectancy is lower even among middle-class people. And once you get to the UK, you have National Health Service, really good healthcare comparatively, and a dramatic reduction in exposure to diseases. And another one of my students, Lauren Houghton, who's working with children, she's actually been collecting things like stool samples. We've been doing parasitology studies, and we know that these middle-class girls, quite a high proportion, do have intestinal parasites. If you look at their health histories, they suffer from a lot more childhood diseases than the girls growing up in Britain. So it's probably disease exposure. And if you look at, take a life history approach and you think about how you allocate resources between growth, maintenance, and reproduction, if you're spending a lot of your energy in fighting off disease as a kid, you'll simply have less energy to put into your reproductive efforts. So we think that probably explains why you have a later age at menopause and possibly these lower levels of hormones. What's going on with menopause? This is fascinating to me, and we're trying to get some money to actually look at some of the ovarian mechanisms that could explain why menopause is pushed later. What's going on there? I don't know the answer to that. I mean, obviously there are these life history, there's a life history overview about energetic availability, but how this is mediated mechanistically is something I'd really like to know more about. Yeah, there's a question at the back. Yeah. Yeah, this is an interesting question. We don't, I mean, the statistics that I gave you come from sort of published statistics in, you know, the Office of National Statistics in Britain. So I don't have access to sort of specific clinical data from hospitals. But the Office of National Statistics is compiling data on populations in Britain. So we've got, you know, the South Asians when they come in, and the South Asians later. I don't think that in those groups, given the sort of general access to clinical care in Britain, which doesn't differentiate by socioeconomics because it's free healthcare, I don't think you're going to see a distinction in sort of how these postmenopausal cancers are being picked up. And I don't know enough about the statistics to be able to answer your other question about what types of cancers they are, but I assume from the age distribution of these cancers that they are hormone receptor positive, estrogen receptor positive types of cancers rather than any other kind. Yeah, question over here.
You mentioned postmenopausal fatness as being associated, and I was wondering
Yes.
about BMI in general and how changes in those rates tracked compared to the increase in breast cancer.
We haven't done a correlation, actually, so that might be something that would be interesting to look at. Yeah. I mean, what will happen with the second generation is quite interesting, too, because my student, Lauren, who's working with the young girls, she's looking at girls aged 5 to 16. A lot of the adolescent girls, the Bangladeshi girls, are desperately dieting all the time and trying to keep their weight low. And if you ask them why, it's because they say that they're quite conservative still about how partners are chosen. I would say the majority are still happy to have their parents pick a husband for them, but their rationale for this is that they'll have better chance of getting a good husband if they're thin, which is interesting culturally. Yeah, question at the back.
I think the concept of hormonal evolution has been pretty well accepted, but it seems like recently the discussion has sort of changed into how not only the tumor cells are genetically altered, but the stromal cells, particularly like in breast cancer, and whether such concept of kin selection applies to cell level in the cancer setting. In other words, it seems quite well accepted that cancer cells evolve, but the mechanism of the evolution is
Yeah, well, it's interesting because the degree of kinship between a cancer cell and a somatic cell is certainly greater than the degree of kinship between a brother and a sister. However, having said that, you've probably noticed that children do get into fights with each other. I think maybe the most important thing here is to note that the degree of kinship is not across the entire genome. The degree of kinship is between genes that are identical by descent. And the genes in a somatic clone of cancer that result from mutations are not identical by descent with the genes in the clones that they are competing with. And if those particular genes, if you just look at one gene at a time, are unrelated to the ones in the other clones, then there's no kin selection issue there. They're independent agents, and they're following their own selfish interest. You know, my two sons are 99.9% identical to each other in the genome, but from the point of view of kin selection, that's not the part of it that's important. The part of it that's important are all of the genes that differ between them because they got different products of meiosis in me and my wife. Okay? This is covered very well in an essay by Alan Graffin that I think it's 1987. And you have to actually think in a fairly nuanced way about what you're talking about when you're talking about relationship.